Hello, everyone, and thank you for the opportunity to present today. I'm here to provide an in-depth overview of the ICH Q3C Guideline for residual solvents, including the scientific rationale behind solvent classification, toxicological thresholds, and the upcoming changes under Q3C, R10. This presentation integrates data from the official ICH guidance documents and support papers, intended for a professional audience involved in pharmaceutical regulation, toxicology, quality assurance, and drug development. ICH Q3C was created to establish a consistent global policy on limiting residual solvents in pharmaceutical products. Residual solvents are organic volatile chemicals that remain from the manufacturing or formulation processes. While necessary in synthesis or purification, they do not provide therapeutic benefit and may pose health risks if not controlled. The guideline outlines PDEs, or permitted daily exposures, that are derived using established toxicological data and exposure models. These thresholds are critical to ensuring drug safety without hindering manufacturing feasibility. The concept of harmonizing solvent limits emerged at ICH2 in 1993. The initial Q3C guideline was finalized in July 1997. Over time, several revisions followed. R1 through R3 incorporated new solvents like THF and NMP. R5 and R6 introduced PDEs for cumene, methylisobutylketone, and triethylamine. R8 added solvents such as 2-MTHF and tertiary butyl alcohol. R9, adopted in January 2024, made technical clarifications and analytical method considerations. We are now approaching the Q3C, R10 phase with substantial toxicological and structural updates. ICH Q3C classifies solvents based on risk. Class 1. These include known human carcinogens like benzene and carbon tetrachloride. They should be avoided unless absolutely essential. Class 2. These solvents, like dichloromethane and DMF, are associated with significant but non-genotoxic or reversible toxicities. Their use is permitted within tight limits. Class 3. These have a well-established safety profile in humans when exposure is less than 50 milligrams per day. Examples include ethanol, acetone, and acetic acid. The risk-based classification stems from long-term toxicological studies. Class 1 solvents show strong carcinogenic potential in humans or animals, or have significant environmental hazards. Class 2 includes solvents with organ toxicity, teratogenicity, or carcinogenicity seen in animals, but with no strong genotoxic mechanism. Class 3 solvents are supported by repeat dose studies and show minimal systemic toxicity. Regulatory concern is minimal if levels remain below 0.5% or 50 milligrams per day. PDEs are calculated using NOEL or LOL values from animal studies, applying uncertainty factors for interspecies variability, duration of study, and severity of endpoint. There are two compliance approaches. Option 1. Apply fixed ppm limits assuming a 10 g/day drug product dose. Option 2. Use actual daily dose and PDE to calculate specific limits per component. Analytical method validation is essential, and nonspecific methods like loss on drying are only acceptable for Class 3 solvents under controlled circumstances. Q3C, R9, was finalized in January 2024. It includes clarified guidance in Section 3.4 on volatility impacts during analysis, reinforcement that analytical methods must capture low boiling solvents efficiently. No major PDE changes, but a revalidation of data confirmed current limits. DMF 8.8 milligrams per day. DCM, 6.0 milligrams per day. ICH, 6.2 milligrams per day. The document also consolidates monographs in appendices 2 to 6, which will be reorganized under R10. The upcoming Q3C R10 introduces revisions based on DMF. IRC upgraded its classification to Group 2, probably carcinogenic to humans, due to liver cancer evidence. DCM, under ECHA proposal, reclassified to CARC 1B and MUTA 2, meaning it may need to move from Class 2 to Class 1. ICH, the 2004 NTP monograph revealed developmental and reproductive toxicity not originally considered. The EWG is assessing these datasets to determine if PDEs should be revised and whether reclassification is justified. The ICH Q3C R10 expert working group initiated the concept paper in late 2024. The following milestones have been set. July 2025, data evaluation and monograph drafting. September 2025, Step 1 sign-off of revised DMF, DCM, EG monographs and structural changes. October 2025, Step 2 A/2B, opening for public consultation. March 2026, Step 3 closure of comments. November 2026, Step 4 final adoption. This ensures transparency and scientific rigor through all stages. Support documents serve as the scientific foundation. Document 1, 2018 summarizes toxicity of Class 1 solvents. Includes detailed animal data and PDE derivations. Document 2, 2020 clarifies the correct PDE for ethylene glycol, 6.2 milligrams per day, fixing discrepancies in older versions. Document 3, 2018 covers Class 3 solvents with toxicological justification for the greater than or equal to 50 milligrams per day threshold. These documents will inform the revised monographs in R10. As outlined in Annex 4, revisions are only considered with significant new or previously unrecognized toxicity data. Proposals may originate from any ICH member, observer, or stakeholder. The EWG, led by a rotating rapporteur, evaluates proposals biannually. Revisions require MC and Assembly approval, followed by public consultation and pharmacopoeia publication. This structured approach ensures consistency, scientific robustness, and stakeholder input. One of the major usability improvements in Q3C R10 is the separation of compound-specific monographs from the core guideline. Currently, monographs are scattered across appendices and support files. In R10, all monographs will be consolidated into one document. They will be directly linked to the solvent tables in the main guideline. This structural clarity improves accessibility for regulators and manufacturers alike. To summarize, ICH Q3C is a cornerstone for ensuring pharmaceutical safety via residual solvent control. The upcoming R10 revision addresses updated carcinogenic and reproductive risk data for DMF, DCM, and ICH. Structural reorganization will make the guideline more user-friendly. I encourage you to follow the public consultation phase and participate in the review process if relevant. Thank you for your attention. I now welcome any questions or comments from the audience.